In the earlier slide, I went through some of the components of the duct system, and what I mentioned was the spermatic ducts. So these are going to be the ducts prior to reaching the urethra. So the efferent ductules, the epididymis, and the ductus deferens were all mentioned in the earlier slide, and the one that was not visible there, but will be mentioned now, is the ejaculatory duct. And that's where the ductus deferens and the duct of the seminal vesicle will meet one another, and that will pass through the prostate and then empty into the urethra. So the ejaculatory duct is going to be the last duct prior to entering that urethra side. And as a reminder, the urethra is going to be a shared pathway with respect to the urinary system as well as the reproductive system, but there will not be simultaneous release of urine and semen as a result of certain processes that we won't discuss in this particular class. But in looking at the urethra, you can also see that it's broken down into several different regions. And this is specific to the male reproductive tract. There's not going to be this distinction that's made within the female reproductive tract. And the urethra in the male is significantly longer when compared to the female. Just for exam purposes, these portions of the penis are not essential. Glans is, because that is mentioned within your book, along with the foreskin area. And then, of course, we would care about the urethral orifice or opening in which the components of semen will exit. Everything else that's here within this image are things that we have discussed as it relates to the testes or duct system. So you're responsible for those things. So just to reiterate, these particular vocabulary terms within this image are not essential. The glans is, because that is something that you already have within the book, and everything else we've already discussed. The purpose of this particular slide is just to provide an area to address the accessory glands. So this slide does have a lot of information on it that is beyond the scope of what you need. So we're just going through what's highlighted in yellow as it relates to this slide. So you can just think about it with respect to where do we find it within the reproductive tract so that you have a view of it, but you're only responsible for what's highlighted in this particular slide. We'll start with the seminal gland, which makes up the bulk of semen volume. The percentage value fluctuates from book to book, but the idea behind the seminal gland is that it will release a kind of alkaline fluid, so a basic fluid that contains the sugar fructose, there's citric acid in there, prostaglandins, and other things that can help to increase sperm motility or fertilizing capability. Now, when it comes to the prostate gland is going to be kind of encircling the urethra, just a little bit below the bladder. And when it comes to the prostate gland, during ejaculation, the smooth muscle within the gland will contract, and that will squeeze the secretions that the prostate gland provides into the urethra, and that happens through several ducts. When it comes to the prostate gland, is associated with sperm activation, and there's a little bit of acidity that's associated with that. It contains a nutrient source within there. It contains some enzymes. It also contains something called prostate-specific antigen, or PSA. And this used to be a readout for prostate cancer because it was thought that elevated levels of PSA was attributed to prostate cancer, but there are a lot of false positives that result from using this as an assay, meaning that there may be elevated levels of PSA, but no actual cancer. And when it comes to prostate cancers, these are pretty slow growing cancers. There's actually someone who used to teach within the department who had prostate cancer, and his options were to wait it out or to seek treatment. And waiting it out is actually a pretty common option when it comes to prostate cancers, just due to how slow they grow and the development later within life. So these aren't things that you normally would expect to see in a younger individual. So that can be an acceptable treatment option. But if the cancer is a little bit more advanced, then perhaps there's surgery that would be involved, or surgery in conjunction with radiation therapy. And As is the case with most cancers, they'll range with respect to their severity. So there can absolutely be prostate cancers that may have metastasized. A common metastatic location for prostate cancer is the bone. But when it comes to this particular type of cancer, testosterone plays a large role. And so there can be drugs that will block production of testosterone that can help to slow the progression of the metastatic version. So I think this is a nice way to connect the content that you learn within cancer as far as metastases, progression, tumorigenesis, as it relates to the reproductive system. The bulbourethral gland will secrete a mucus that helps to neutralize any acidic urine that may be left behind within the urethra. And in addition, the mucus can help to lubricate the glans penis during sexual excitation. So that's a level of detail that you need for these three different glands and their overall contribution to semen production. While there are similarities with the male and female reproductive anatomy and physiology, there are some differences with slightly increased complexity with respect to anatomy and physiology of the female system. Now, in addition to gamete production, the process of oogenesis that occurs within the female reproductive system, there's also the need to prepare an environment that's conducive for successful fetal development, a process which takes approximately 40 weeks to occur. Now, when it comes to the female reproductive anatomy, we're going to truncate the list of things to know and really just focus on the internal genitalia. So the internal genitalia is going to include the ovaries and the duct system. So thinking about the oviduct or uterine tubes, the uterus, and the vagina. So there are things shown within this particular image that constitute the external genitalia, but those will not be things that you'll be tested on. The ovaries constitute areas of egg production, and these are the female gonads. And when looking at the ovary cortex, there are tons of little structures found within there that are ovarian follicles. And each follicle consists of an immature egg called an oocyte. And they will be encased by layers of different cells, and they'll go through a process of changes as they develop. And so there will be the follicles that are found within the ovary that vary with respect to where they're at within development. And each month, one of the follicles will develop to a state in which it can be ejected. And that is the process of ovulation. The oviduct or uterine tubes will receive the oocyte that gets released from the ovary, and the oocyte will travel and make its way along the length of the oviduct until it reaches the uterus. The uterus is a muscular organ, and its role is to be a site for receiving a fertilized egg and for providing an area where that fertilized egg can be nourished. When it comes to the uterus, it starts off relatively small but increases in size after pregnancy. So it never reverts quite back to the original size postpartum. When it comes to the uterus, there is a uterine wall, and it's important to keep in mind that the uterine wall is actually composed of more layers than what's shown here. This particular image is only focusing on the endometrium, but there's also the myometrium as well as the perimetrium. But the endometrium that is indicated here is going to be the portion of the uterus that gets shed during menstruation, but it's also the site of implantation if a successful fertilization event occurs. Now, the other one that I'll mention because it will be relevant for our later discussion is the myometrium, which is actually the bulkiest of the layers. So if we're looking at this, all of that is myometrium. That's the portion that's going to contract during childbirth. The cervix is a portion of the uterus, an area in which it kind of narrows and projects into the vagina. And when it comes to the cervix, something that I would like you to keep in mind is that there are mucosa that are present within the cervix that can help to secrete mucus, and that mucus fills the cervical canal, and that plays a role in blocking the spread of bacteria from the vagina into the uterus. It can also block sperm entry apart from specific times within the cycle, and the viscosity of it can change in order to permit sperm to pass through. So we'll think about the idea or relationship between these events and cycles in a later portion of the voice thread. The vagina is going to be the site for menstrual discharge. It is going to be a site for reception of penis and semen during copulation, and it can also serve as an outlet for the birth of a baby. Another thing to keep in mind when it comes to this component of the female reproductive anatomy is that there is microbiota that are present here. And as a result of the microbiota, there's the formation of lactic acid that occurs, and that makes the pH in this location relatively acidic. And the microbiota that are here will benefit the host. So this is something that the morning class has discussed, and I'm sure the evening class as well, with respect to the immune system content. So the relationship between microbiota and the host. And there is going to be a low pH here, which helps to keep the vagina healthy and serves as a deterrent to infection by bacteria that aren't suited for the environmental conditions that are seen here. While the mammary glands are not technically part of the reproductive system, they are an integral part of mammalian reproduction. So we'll go through this visually here. So each mammary gland, and I'm going to focus on the lactating mammary gland below, has lobes, and those lobes will essentially radiate or surround the opening that's at the nipple here. There are suspensory ligaments that are found within the breast tissue, and this is connective tissue that helps to attach the breast to the underlying muscle as well as the overlying dermis. The lobes will contain lobules, and lobules will have milk-producing alveolus or alveoli plural, and this will occur during lactation. And within this process, there will be milk that passes through the lactiferous ducts, which will open to the outside. And then there's also a lactiferous sinus, and this is where milk will accumulate during nursing. So this description is something that applies to those that are nursing or those that are in the last trimester of pregnancy. But otherwise, the organization looks more like what we see up here. And that is largely the fact that the glandular structure of the breast is undeveloped and the duct system is rudimentary. So when it comes to this particular image, largely the size of breast tissue will be based on the presence of adipose tissue. And we've gone over pretty much all of the vocabulary that's here. The only thing I didn't explicitly mention was the epithelium that you see here and here. So I'm not going to expand on it any more than what I've already gone through for this slide. So those will be two things that I'm not going to expect of you very specifically as it relates to this image. Gametogenesis is the formation or production of gametes. So spermatogenesis, more specifically, is the ability to generate sperm. Now, we will make a distinction between spermatogenesis and spermiogenesis, and we'll go through both processes within the voice threads. But when it comes to spermatogenesis, here they're generalizing it in the formation of sperm. But again, just to reiterate, we are going to be really specific as to the distinction between the different processes. One is about the process of getting us towards sperm, whereas spermiogenesis is the maturation of the spermatids that result from the gametogenic events that take place before that. But when it comes to spermatogenesis/spermiogenesis, both of those events, what ends up happening is a really vast amount of sperm that can be produced. So when it comes to the process of sperm production versus sperm development and its ability to be utilized for the process of fertilization, those are two different things. So production of sperm can be short, but then as far as sperm development goes, as you can see here, it can take several weeks in order for proper development to occur. So if we were to take a cross section of the area in which the process of spermatogenesis takes place, we would see various forms of these gametes as they progress towards their ability to be, a fully developed sperm ready to set off on course to find an egg in order to achieve fertilization. This image really is just to provide an opportunity to review these two concepts if there is anything that you have forgotten between the time that you initially learned it and where we are at right now, because certain attributes of these processes are relevant for our discussion of reproduction. So we'll go through mitosis, and then afterwards we'll go through meiosis. Now for meiosis, we're starting with a parental cell that's 2n equals 4. So we are diploid at this point with a total of 4 chromosomes, 2 distinct sets because n is equivalent to 2. And the distinction here, while it is very subtle, can be seen when comparing the lengths of the chromosomes. So these chromosomes are homologous to one another, and these chromosomes are homologous to one another. Now, what will take place prior to entering mitosis is chromosome replication that takes place during S phase. And when we are at prophase, we're still 2n equals 4 here, but we've doubled our genetic content, or the amount of chromatin that's seen within the cell. So for mitotic division, this is something that we describe as equational, because our parental cell is going to be a reflection of what we will ultimately see within our daughter cells. So in mitosis, we will take those duplicated chromosomes consisting of our sister chromatids. They will line up at the equatorial plate during metaphase, and then through the depolymerization of the mitotic spindle and utilization of motor proteins, those sister chromatids will separate, migrate to opposite poles, become their own chromosomes, and eventually be partitioned into 2 distinct cells. So at the end of mitosis, we generate 2 daughter cells. Their ploidy is the same as that of the parent cell. So we start off with 2n equals 4 in the parent cell. We end with 2n equals 4 in each of these daughter cells. So mitosis is an equational division. We prepare for that equational division through chromosome replication and through 1 round of division. In meiosis, we start off the same way as mitosis, which is taking that parental cell and going through chromosome replication. Only in this scenario, we are going to have tetrads. And so those homologous chromosomes will interact with one another. There will be a synapsis of those homologous chromosomes, and that provides an opportunity for the exchange of genetic information to take place. So that exchange of genetic information does not occur during mitosis, but it does during meiosis, or at least the opportunity for that event to take place is present at meiosis. Now, when it comes to meiosis, this is a reductional division. There will be 2 successive rounds of division that occurs here in contrast to 1. So when we are at metaphase 1, rather than having our chromosomes lining up at the metaphase plate, we will have those tetrads that will line up at the metaphase plate, and they'll be held together through that exchange of information that will be occurring. Now, at the end of the first round of meiosis, there is going to be a separation of the dyads that are being found within the tetrad, and those will get partitioned off into their daughter cells. At this point, we are haploid, because when we take a look within the daughter cells that result from meiosis 1, we find that n is equal to 2. So there are 2 distinct types of chromosomes found within here. They are not homologous to one another. Each one of these daughter cells will go through another round of division in order to partition out those sister chromatids. So the second wave of meiosis is comparable to the first of mitosis, in that the sister chromatids will separate and they will become chromosomes. Now, at the end of meiosis 2, we have 4 daughter cells. All of these daughter cells are haploid, so all of these are n is equal to 2 based on our starting parent cell. And within this process, we want to start to make distinctions as it relates to the reproductive system, as when is there mitotic division taking place? When are we desiring something that is going to be maintaining the ploidy and not changing any of the information, versus when we go through meiotic division? So this is really an overview. These are concepts that you've already learned in a really significant way throughout your duration within Principles of Biology, but it's nice to just revisit them as it relates to gametogenesis and the events that allow us to generate our sperm cells and our egg cells. Now we'll go to your second question. So you're asking, are all of them from the same chromosome, and red is from mother and blue is from father? So books will always use blue to represent paternal and red to represent maternal. That's kind of consistent regardless of whether it's Pearson or another textbook at another institution. So when we're taking a look at it from meiosis, because you say here the blue is crossing over with the red, so they can't be from different chromosomes, you are correct in the sense that they are homologous chromosomes. So when we are considering homology here, what you'll reference with respect to the chromosomes themselves, and this kind of answers your third portion, is the length. So we're looking for lengths that are identical. So this would be a maternally derived chromosome from the mom. This would be a paternally derived chromosome, and they're homologous to one another in the same way that these are homologous to one another. And it's very subtle in this image, but I am utilizing their length as a way of indicating which ones are homologous. So you have homologous chromosomes, and that might be what you're saying when you say the same chromosome. So this blue one, just to repeat, this blue one is homologous to this red one. This blue one is homologous to this red one. And when you have crossing over, which we see within metaphase here, you are exchanging material between maternally derived chromosomes and paternally derived chromosomes. They've gone through replication already, so you have those sister chromatids present there as well. So when we're looking at this, the kind of idea behind doing the color discrimination is to help track maternal and paternally derived genetic information, and how we distinguish homologous chromosomes from non-homologous chromosomes would, yes, be based on its length. If there's anything else within what I said, within questions have here that needs